My pleasure to invite up to the podium Professor Rocio Macias from University of Salamanca to talk about transport proteins, key players in cholangiocarcinoma diagnosis, prognosis and treatment.
Thank you so much. Thank you again for the opportunity to present some of the work we do in the EBFAR group in Salamanca related to transport proteins, and I will try to show you why they can be important in diagnosis, prognosis, and treatment. As you know, cells have transport proteins that participate in the entrance or the uptake of compounds they need for their normal functioning. And these transporters belong to the solute carrier family. And they also have other transport proteins that participate in the export of substances the cells don't need or even that can be toxic for them. And these transport proteins belong to the ABC or ADP binding cassette family of proteins and use energy to expel these compounds out of the cells. Some of these transport proteins are ubiquitous, while others are expressed in some tissues, and in some cases in one tissue we have one type of transporters only in one type of cells. And this type of transport proteins expressed in one cell type are of special interest in diagnosis. And this is because in tumor cells, the transport proteins we find are similar to those that are in the healthiest tissue. And we can find in the tumor cells changes in the expression or variations in genetic or epigenetic alterations. Transport proteins that participate in the uptake of bile acids meet the requirements for the diagnosis. For instance, the apical sodium bile salt transporter ASBT is located only in cholangiocytes and not in hepatocytes. And the NTCP, the sodium liposotransporting polypeptide, is only expressed in hepatocytes. And this could be used in the tumors to differentiate intrahepatic cholangiocarcinoma and hepatocellular carcinoma. But there are other transport proteins with similar characteristics, like the glucose transporter expressed in cholangiocytes, or the aquaporin-1 expressed also in cholangiocytes, with high levels in cholangiocarcinoma and very low levels in hepatocellular carcinoma. I will comment a little more about ASBT. This transport protein is expressed in the intestine and permit the uptake of bile acids and back to the liver and the maintenance of the enterohepatic circulation. It's also expressed in the renal tubule, where this transporter can take up bile acids that are filtered in the kidney. But in the liver, which is interesting, is that it's only expressed in cholangiocytes. And when we analyze the expression of these transport proteins in cell lines derived of epithelial, biliary epithelial, we found that most of the cell lines express moderately this transport protein, even with higher levels than normal cholangiocytes, while this was not expressed in hepatocellular carcinoma cell lines. And when we analyzed in tumors, we found moderate expression in cholangiocarcinomas and was not detected in hepatocellular carcinoma. Here you can see that this high expression in cholangiocarcinoma is very heterogeneous. We have some cases with a very good expression and others with a lower expression. And we can see that while in normal cells we have the expression in the apical membrane, in the tumor cells that lost polarization, they are expressed in the plasma membrane. Even if not all the tumors have this ABC transporter, the heterogeneity could be solved using a panel of biomarkers. But the presence of this transport protein in certain cholangiocarcinomas could be useful for other applications, as I will show you later. Another thing you know is that there are different mechanisms of chemoresistance. In addition to one related to transport proteins that I will mention later, we have alterations in the enzymes involved in drug metabolism, changes in the molecular targets, an enhanced DNA repair when we have drugs that bind to the DNA, an enhanced survival of or survival pathways and decreased apoptosis, and changes in the tumor microenvironment that reduce the effect of the drugs, and also an enhanced phenotype transition from epithelial to mesenchymal, and we know that the mesenchymal cells are highly resistant. So related to these transport proteins, we know that a reduced uptake of the drug or an increased expulsion of these anti-cancer agents will lead to a reduction of the intracellular concentration of this compound and then to a lower effect. So the analysis of the transportome, which is defined as the set of transporters expressed in the cancer cells at a certain moment, can be part of the complex picture that can justify the response or not response to certain pharmacological agents. And here I will show you some transport proteins that have been associated with response to the treatment. For instance, the nucleoside transporter N1, when it's present, has been associated with response to gemcitabine. Or the organic cation transported 1, OCT1, has been associated with response to sorafenib. Or the copper transporter CTR1 associated with response to platinum derivatives. I will show you some examples, but here you can see two pictures of two tumors, one with a high expression of N1 and another with no expression. And this study showed that the presence of N1 was associated with a higher survival. Later, what was demonstrated was that it was not important to have N1, but to have N1 in the plasma membrane. Since this is a transport protein that must be localized in the plasma membrane, if we have it in the cytoplasm, it's not going to participate in this transport protein. And you can see how the survival was higher in the patients with tumors that have this transport protein in the plasma membrane compared with the absence in patients treated with adjuvant gemcitabine. We found similar results analyzing the transport of sorafenib. We found that sorafenib is transported by OCT1, and we found that it was very frequent to have mutations in these transport proteins in hepatocellular carcinoma. So what we did was to induce the expression of, in one cell line derived from cholangiocarcinoma, of OCT1, and we could see that these cells were able to take up sorafenib. And when, I will skip it, and when they cells take up sorafenib, they have a reduction in the cell viability. And we found mutations in this transport protein. We expressed these mutated proteins in cells, and we could demonstrate that some of them were not able to transport sorafenib. And in fact, the protein was localized in the intracellular part of the cells, instead that in the membrane, and the uptake was reduced. We performed immunohistochemistry in tumors, in hepatocellular carcinoma tumors, and found that half of the tumors had no OCT1, while the other part, we could detect these transport proteins. But there were no differences in the survival saying yes or no positivity of OCT1. However, when considering only the presence of OCT1 in the plasma membrane, we found a similar result with OCT1 presence associated with higher overall survival. What we found surprising was that some cases without OCT1 were among patients with a higher survival. We couldn't explain that until recently, in which when we could demonstrate that there is another transport protein that is not as efficiently good transported, but can also transport sorafenib. We expressed cells with OCT3, and we could demonstrate that they were able also to take up sorafenib. In fact, in these experiments, we could see that maybe, because this is an indirect way to show that this transporter may be also transport regorafenib, lenvatinib, cabozantinib in addition to sorafenib. And we did the same experiments in the same patients of detecting OCT3. We found similar, some negative cases, very low expression, moderate, high, very clear expression in the plasma membrane, and we found a similar result. When we had OCT3 in the plasma membrane, the survival after treatment with sorafenib was higher. And combining the presence of one of them, the significance was even higher. So sorafenib is not used for cholangiocarcinoma. You know, it's a tyrosine kinase inhibitor, and that was used for many years in hepatocellular carcinoma. Now we have immunotherapy with better results. But this drug is not working in cholangiocarcinoma, at least not in the majority of patients. It's not used. But in some cases that were misdiagnosis, was having some effects. So what we found is that this transport protein is not expressed in cholangiocarcinomas. We check in different cohorts, and this was associated with hypermethylation of the promoter. And we use cholangiocarcinoma cell lines, 2 derived from extrahepatic and 1 from intrahepatic, and we induce the expression of OCT1. You can see here that we have high levels by quantitative PCR of OCT1 in all of the cells. And when this is expressed, they have the ability to take up a substrate of OCT1. They are able to take up sorafenib, and we have a reduction in the viability of the cells. So we designed a gene therapy strategy to overexpress OCT1 in vivo. We use, in this case, adenoviruses, and we express, and we designed to have OCT1 under the control of the sorvivin promoter. We intended that OCT1 would be only expressed in the presence of sorvivin. And sorvivin, as you know, is an anti-apoptotic protein that is only present in tumor cells, but not in normal cells specialized. So what we did was we used a donor mouse to inject cholangiocarcinoma cells. When we had a tumor, we used this tumor to obtain small fragments that were introduced in the liver of receptor mice, and we divided into 4 groups. One group receiving this adenovirus without OCT1 and the vehicle, another one receiving sorafenib, and then 2 other groups receiving the adenoviruses with OCT1, and one of them was treated with sorafenib, and the other one with the vehicle. And you can see here that the administration of adenoviruses making the expression of OCT1 and the treatment with sorafenib significantly reduced the growth of the tumors in these animals. So this is important because we have to keep in mind that not these drugs, but all type of drugs, including targeted drugs, use transport proteins to reach their molecular targets. I mean, ivosidenib needs to bind intracellular enzymes mutated, but these drugs that bind to receptors bind the internal part of the receptor, so they need to go inside the cells. And we know that even in studies in which the patients had been selected and have this FGFR2 fusion or rearrangement, in this case with pemigatinib or futibatinib, not all the patients are going to respond. So maybe there are other aspects that needs to be considered to know if they are going to respond. So we are characterizing the transport proteins involved in the transport of these drugs, and we can see that there are very important differences in the expression of some of these transporters that participate in this movement of drugs. I will switch to other opportunities to use these transport proteins, and there are some that have been associated directly with prognosis. For instance, the presence of the glucose transporter 2 has been associated with high-grade biliary tumors by immunohistochemistry. Phospholipolene is a marker of a favorable outcome, and the presence of these export pumps have been associated with reduced or low prognosis, even when the patients are not treated with drugs that are transported by these transport proteins. And then the other thing that we can use, knowing more about these transport proteins, is that we can either modify the expression, or we can use the presence of certain transport protein to deliver drugs. So for instance, we know this ASBT is able to transport bile acids, and we have synthesized bile acid derivatives to have the drug inside the cells in higher amounts. Or we know that the presence of OCT1 can be altered in presence of the decitabine and cisplatin. And one also can be overexpressed when the cells have been previously treated with cisplatin. So we can know more about how we can modulate the expression of transport proteins to do an organized administration of drugs. Just to illustrate this part I was telling you, we know that ASBT is in cholangiocytes, and in some cases in which we don't have transport proteins for cisplatin, for instance, we don't have this CTR1, and we know that we have ASBT. What we have done is to put a cisplatin together with bile acids. This is one of the compounds we have synthesized, BAMET-UD2. Is cisplatin with 2 molecules of ursodeoxycholic acid. And what we see is that when we express in the cells ASBT and we treat with cisplatin, we have an effect. But when we treat it with BAMET-UD2, we have a higher effect. And we can see that the entrance is mediated by ASBT, because when we use, we incubate together BAMET-UD2 with a ursodeoxycholic acid, a bile acid, we block completely the effect of this compound. So another thing that is interesting with these compounds is that they are maintained for a certain time in the enterohepatic circulation. That means that we reduce the side effects of the cisplatin, for instance, because we have lower amounts in the general circulation, and we need lower amounts of compound because it's going to be maintained in this circulation. We use animals with cholangiocarcinoma induced by administration of thioacetamide in the water, and we were able to show that these tumors maintain the ability to take up a fluorescent bile acid derivative, FIGC. You see here the colocalization with cytokeratin 7. And again, when we check in these animals after one time of the administration, the presence of platinum in the serum, the levels were higher with cisplatin and very low with BAMET-UD2, and the tissue serum ratio was higher in the tumors after administering BAMET-UD2. So again, we can use these transport proteins and modulate the expression if we know more about also the export pumps, and we can use the administration of another compound together with our drug to increase the effect. So in summary, I will just give you 3 conclusions. These transport proteins can be used for screening. Certain transport proteins are expressed only in cholangiocytes and have the potential to help in the diagnosis of cholangiocarcinoma. Some can be used to predict response. If proteins involved in the transport of each drug are well characterized. The transcriptome analysis can help to predict the lack of response to a certain treatment. And we can use this information for chemosensitization. In addition to new and more effective drugs, we can combine different strategies to chemosensitize using the tools we have. And with that, we can improve the rates of drug treatments to cholangiocarcinoma. And I just want to thank all the members of my group and the collaborators, and thank you very much for your attention.